today we're going to be talking about DNA replication. Yay! So this video is going to come in two parts because it's a little bit long if I make it all in one part. So right now we're going to focus on what happens up to the replication fork. And then we're also going to focus on the leading strand synthesis. And then in the next video, we'll review really quickly what happens leading up to the replication fork. And we'll also go over what happens to the lagging strand during replication. So first things first, this is going to be more of an in-depth overview than you may need for any of your introductory level botany classes or biology classes or both, honestly. So just be prepared. This is a lot of information, but I will try to simplify it and make it as easy to understand as possible. So with that said, let's start talking about how we begin DNA replication. So if you look at the top here, there's an orange little blob that is topoisomerase, or otherwise known as DNA gyrase. So this is responsible for keeping the DNA from supercoiling. What that basically means is that we need DNA to be in a format where it's ready to split here at this replication fork. We need DNA to be not coiled in on itself, not super condensed or compact, so that we can split it apart here. So topoisomerase is going to be responsible for keeping that DNA from being really compact, from supercoiling. So as you can see, there are two strands here of what are called parent DNA, and this is going to all split here at this central location. That location is called the replication fork. I'll go over that a little bit more in detail in some other videos, or reference it in some other videos as well, still looking at DNA replication, but all you need to know for now is that the area where these two parent strands are going to split off is the replication fork. Now these will split off into the leading and lagging strands. Like I said, we're going to talk mostly about the leading strand here today for this video, and part 2 will concern the lagging strand here. So with that in mind, we're going to kind of cut it off here and skip around this way. So following this light blue strand here, this light blue parent strand, which is part of the original DNA molecule, we're looking at some of these light purple dots. All of those light purple dots are SSBs, single-stranded binding proteins. Now, what are those? It's exactly what they're called. These are single-stranded binding, so they're binding to the single strand of DNA, and they're proteins. So the function of these is going to be to keep the single strand from winding back in on itself or wanting to bind back with its other strand, with its other dark blue parent strand over here. Because after we go through DNA helicase, which unwinds or unzips the double-stranded DNA, DNA wants to be in this double-stranded format. Whenever we think of DNA, we think of, right, this like kind of this double-stranded, this helix. That's because DNA wants to be together. It wants to be complementarily bound to its partner. So helicase is going to be responsible for unwinding that double strand. Once we unwind that double strand, we need these single-stranded binding proteins to keep those two strands from coming right back together again. It's going to want to go like this, right, and continue to be double-stranded all the way. But no, these single-stranded binding proteins will kind of temporarily satisfy that single strand of DNA until it gets a new partner, which is in teal, and that's the newly synthesized strand there. So there we have topoisomerase keeping the DNA ready to be unwound, DNA helicase, which is unzipping the double strand, and the single-stranded binding proteins, which are helping the strands to not come back together again. You'll notice everything up to this point is just mainly about keeping that DNA happy until we can get it a new partner here. So up to this point, we're looking at right here, DNA polymerase III. That's this big pink blob there. So there's a few different types of DNA polymerase. As you can see on the left, there's DNA polymerase III and DNA polymerase I. DNA polymerase III is mostly responsible for synthesizing the new strand of DNA. So that's this teal here. So DNA pol 3 is going to do the same thing on this left strand, on this leading strand, and on this right strand or the lagging strand. Okay, next we're going to discuss the beta clamp. Beta clamp, you probably don't need to know that much about it, but the purpose of that is to stabilize the strand of parent DNA and the strand of newly synthesized DNA. You can see the parent strand is in light blue, and the newly synthesized strand is in this green tealish color right here. So that's all about the leading strand. I'm going to break this video up, like I said, into 2 parts. So I'm going to link to the next video about the lagging strand synthesis, and I will cover that in the next video. If you have anything that you want me to cover or any suggestions or recommendations, please let me know in the comments and let me know if you enjoyed this video or found it helpful at all. Alright, I will see you on part 2.